Okay, so now we've seen glycolysis in its entirety. And the question then is, what next? What do we do with it next? Well, that's what I want to get into now, and I want to take us back. We're going to go back to some of the earliest discoveries that led to all of these questions in the first place. And they started with this gentleman here, William Harvey. Harvey was an English scientist, biologist, physiologist, very, very long ago, back in the 17th century. And he is the first person to discover really what the heart and circulatory system were doing. Now, it had already been known that the heart sort of moved the blood around. It wasn't really well understood, though, exactly what was going on with the cardiovascular system, whether or not the blood just sort of sloshed around or was somehow mixed in the heart or something like this. And Harvey did a series of really beautiful experiments that showed essentially what the heart and blood vessels are doing. What he showed, first of all, was that the heart does not mix the blood. It was thought that maybe that was the case by one of the hypotheses. And what he did is he took an ox heart and he filled it full of water, and he showed that the water on one side of the heart does not mix with the other side, because he filled only one half of the heart with water, and it never went through. He also did a series of really clever experiments by putting clamps in various locations on various blood vessels, and showed that on the vessels that were bluish, if you put a clamp on it, they would become empty of blood towards the heart and fill up and distend and get full away from the heart. And then the red ones, if you clamp those would get full towards the heart and empty away from the heart. So what he was basically showing was that there was a circulatory system. And then he also showed in these pictures here that you see, he showed that in your hand and arm, the veins have valves, and they're one-way valves. Now, people knew the valves were there, but they didn't know what their function was. And he showed that their function was to keep the blood flowing in one direction. So he is really the beginning of this whole process, because like I said before, that marshmallow is connected to your cardiovascular system in a very, very important way. Your cardiovascular system exists because you eat things like marshmallows. Now, what's the relationship between these things? Well, the cardiovascular system is connected to this in the following way. Through a series of experiments done by this another Englishman, Joseph Priestley. Now, at the same time, another French scientist that was working at the same time basically made the same discovery. We think it's hard to tell. They were working at the same time. They communicated to each other, and it wasn't quite clear whether or not Lavoisier, who's the French guy, actually saw these things at the same time, or whether he got it from Joseph Priestley. Of course, the British all believe it was Priestley that did all the work, and the French all believe it was Lavoisier, but whatever. I'm going to show you Priestley's experiments. What Priestley did was he showed that if you put in a closed container, and he put the container was closed by having basically a big open jar that he put the mouth underwater, and he could completely seal it with water. If he put a mouse in there, he would put a little platform in there for the mouse, and another candle, the candle he would stick in from the top with a cork. If he put those two things into a chamber, then something very interesting happened. The moment that the candle went out, the mouse would die. And you could do it either way. You could do it with just the candle, in which case it would last longer, but it would eventually go out. And you could do it with a mouse too. And again, the mouse would last longer, but it would eventually go out. But the key observation was that the mouse and the candle were going out at the same time. So something is going on that's happening here. And so what he then concludes after a series of experiments, this is a very, very great simplification of all the experiments that he did. But what that basically tells him is that the process of burning that's occurring in the candle is related in a very, very deep and important way to what's going on inside the mouse. Now remember, a candle burning is oxidation. So this is the first real discovery, although he didn't state it in these terms. This is the first real discovery that what's going on inside of us, inside of that mouse, is oxidation. It's the same as a burning candle. And something in there is required in order to allow that oxidation to occur. And that thing is replenished by plants, because in a series of other experiments, what he did is he took mint, growing mint, put it into the chamber. Now, he didn't actually have it in a pot, but he would put a sprig in that was still growing. And what he showed was that by putting that in, as long as that sprig was there and in good shape, both the candle and the mouse continued to survive. So this is showing something. Not just the discovery. This is often how it's expressed. This is the discovery of oxygen. Well, yeah, it kind of is. Oxygen had already sort of been known, but what he did was he showed that there was this gas in the atmosphere that is required for oxidation, and this oxidation of the candle is the same as what's going on inside the mouse. That's what's important, really. And it's related then to the cardiovascular system in this way. It's related because we need that gas, we need that oxygen in order to oxidize. So what happens is this: all creatures that use oxygen in their metabolism, including human beings, will take oxygen out of the environment in some way, either directly out of the atmosphere or in water, if you're a fish, for example. Then there'd be a respiratory structure, some surface. In us, it's the lungs, and in fish, it's their gills. And the oxygen diffuses across that surface and into the blood. The blood then carries it throughout the body, where it's absorbed by the cells. And then the cells absorb that oxygen, use it for something, and then in the process produce carbon dioxide, which then goes back into the blood, back to the respiratory surface, and then this carbon dioxide is released. Okay, so oxygen then is carried from the external environment to the tissues, and the tissues are producing carbon dioxide and releasing that into the environment. Okay, which is why it is you have lungs. You breathe in oxygen, you breathe out CO2. Okay, so the question then is why? Right now, you may or may not know why. One of the things that we know that they didn't was that it isn't the entire cell that's using it. It's only one structure inside the cell that's really using that oxygen and producing the carbon dioxide, and it's the mitochondria. So let's go back though, before our understanding, before our real understanding of what the mitochondria were doing. Why is it that the O2 is being carried to cells, and why is it that the CO2 is being released? Okay, well, we've already seen this. We already saw. If you eat a marshmallow, it's got fructose in it. The fructose then gets oxidized. You need oxygen to balance the equation. The equation produces CO2. It makes sense, no problem. But how then? How does it do that? And what is it the mitochondria are actually doing in order to actually work that out? Even before mitochondria were understood, it was worked out what is actually happening with all of that. Now, the issue then comes to this. Here's what we know. We know glycolysis, okay? So we've already looked at this. And in glycolysis, we see glucose goes glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-bisphosphate, DHAP and G3P, and then 1,3-bisphosphoglycerate, then 3PG, 2PG, PEP, and pyruvate. We've seen all of that. Okay, let's go back to this though. How does it connect to this? Where's the oxygen being used? Show me in this reaction scheme, where is oxygen used? There's not one step, not one, that uses oxygen. Okay, where's the CO2 being produced? Again, show me where. Not one step produces CO2. There's a step that produces water, but there's no step that produces CO2. So glycolysis cannot explain how it is that the CO2 is being produced and the oxygen is being used. So there must be more than glycolysis going on. And in fact, that makes sense because glycolysis really isn't producing a lot of ATP. It produces 2 ATP per glucose a net, and the pyruvate is not completely oxidized. So the question remains, where are these gases being consumed and produced? It's not glycolysis. So what could it be? So most of this was worked out in the 1920s, 30s, and 40s. And if we look at this situation right here, in 1920, 1930, this was known. This was understood. So the conundrum I just expressed was understood back then. And people trying to figure out what exactly was going on had figured out ways of measuring how oxygen is consumed and CO2 is produced and other gases and so forth produced during respiration. I'll show you how that works here in just a little bit. In some of their experiments, this is a summary of the key discoveries. Here, if we look at tissue, now here's what we're going to do. Imagine this. I'm going to make a chamber, and inside that chamber, I'm going to put a bunch of living cells. And those cells I'm going to feed various things. I can feed them sugar, I can feed them whatever. Now I know this. I know if I feed cells sugar, they consume oxygen and produce CO2. That was known in the 1920s, 1930s, no big deal. But they couldn't connect it to glycolysis. They already knew that the sugar went into the glycolytic pathway. But the other thing is then, if we don't do anything, we just sort of feed it naturally, this is what happens. We get a bunch of oxygen being consumed right here. And again, on this axis, you're looking at the oxygen consumption rate on the blue bars, and that's in micromoles per gram per hour, micromoles per gram of tissue per hour. And this is producing that much oxygen if you don't do anything to the cells except put it in the chamber, give it sugar, and let it live. Now, if you also measure the carbon dioxide that's being produced, you don't really get a measurable amount of carbon dioxide coming out, interestingly enough. The reason for that is the carbon dioxide goes into, there's so little carbon dioxide being produced, it goes into solution in the water as carbonic acid, and you can't actually see it in your chamber. Okay, but then something interesting happens. We know you feed it sugar, blah, 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 but what if you feed it pyruvate? Remember, pyruvate is the conjugate base of an acid, and it's at the end of glycolysis. If you feed a pyruvate, this happens. Well, look at that. You tell me what happened. Don't let me tell you. You tell me. What happened to the oxygen consumption? What happened to the CO2 production? You can see both went up. So, in other words, if you give cells pyruvate, they start producing more carbon dioxide, and they start absorbing more oxygen. So, people were trying all different sorts of things. And they said, okay, well, pyruvate is an acid, an organic acid. Let's try different organic acids. And so they tried vinegar, acetic acid. And in fact, if they put in acetate, which is the conjugate base of acetic acid, look what happened. The CO2 production goes up even more, and oxygen consumption goes up even more. Okay, so let's try others. There's one called succinic acid. So they put in the conjugate base of it, succinate, and even more CO2 is produced and more oxygen is consumed. Another one, fumarate, fumaric acid. Same thing. Malate, malic acid, same thing. All of these are organic acids. And by feeding cells organic acids, it causes their oxygen consumption to go up and their CO2 production to go up. So what that tells us is that there's something going on that's not glycolysis, and whatever it is, it has something to do with organic acids. How in the world? That was a total, just came out of nowhere. People did not expect organic acids to be important. We've expected carbohydrates to be important, not carboxyl groups. So what's going on with this? Well, one of the things that was known at that time is that all of these are very similar in structure. They all have very similar types of structures. Like I said, they all have a carboxyl group. Remember, organic acids are generally have carboxyl groups. And so this tells us that these organic acids that are all related to each other in some way have something to do with respiration. All right, so if we look at these, succinate, fumarate, malate, and we go back to the 1920s, 1930s, we find out that it was known by, at least suspected anyway, by Thunberg, that those molecules are related in the following way. Notice, here's succinate. Notice what it's got here. We got two central carbons that just have hydrogens on them. And then we have a carboxyl group on this end and a carboxyl group on this end. Those are the acids. So this is what we call a dicarboxylic acid because it has two carboxyls. Now, notice how similar it is to fumarate. Fumarate is exactly the same, except instead of having two hydrogens here, there's a double bond and the hydrogens are gone. And what Thunberg had suspected and or showed by that time was that you can actually convert succinate to fumarate by removing two hydrogens, reflecting those open valences back on the carbons and make this bond here. And in the process, though, you can take those hydrogens and use those to reduce something else. And he didn't discover this, I don't think. Later it was discovered that there's an enzyme, succinate dehydrogenase, that does exactly that. And it takes as a cofactor this thing called FAD. And that is like NAD. It's an adenine dinucleotide. It's just got a different sort of thing besides nicotinamide. But notice it has the same sort of process. It grabs these two hydrogens, and so therefore it's carrying two electrons as well. So this is a reduction step. Delta G therefore must be positive, which means succinate to fumarate must have a negative delta G. So this is exergonic to drive this endergonic reaction. And if you look at fumarate, it's also very similar to malate. In fact, malate, succinate, and fumarate are almost all identical. The only difference between succinate and malate is the oxygen right here. And that's, again, very similar to fumarate. And it was then later showed, again, Thunberg had suggested that this was possible, but it was later showed that there is an enzyme called fumarase, which will take water, split it, open this bond here, replace this hydrogen here, and take the OH and put it here to make malate. So you can make succinate into fumarate into malate. And so again, that fits very well here, succinate fumarate malate. That's why all of these increase CO2 production and oxygen consumption, because they're all very similar molecules. So somehow this is related to oxygen metabolism. But there's something else, and this is also suggested by Thunberg, that malate is very similar to this molecule, oxaloacetate, which is the conjugate base of oxaloacetic acid. And the difference is only this. Notice carboxyl group's the same. This is all the same. The only difference is this carbon right here, the two hydrogens are gone, and there is a double bond with the oxygen. And what was later showed was that this malate can be converted to oxaloacetate by removing these two hydrogens. Now again, removing hydrogens is oxidation. So we're oxidizing malate to oxaloacetate, releasing energy, and the energy then is captured by our old friend NAD, nicotinamide adenine dinucleotide. Again, these two hydrogens take their electrons and put it on NADH. So NADH then is carrying two high energy electrons here. So therefore, I'm going to ask you this. What would you predict? We know what happens when you put succinate, fumarate, and malate into cells. What would you predict if you put oxaloacetate into cells? Would the CO2 go up or down? Would the oxygen consumption go up or down? Well, you would predict, one would hope, that the succinate, fumarate, malate, and oxaloacetate would all act the same way. And in fact, they discovered later, in using the same experiments that I showed you earlier, that that's exactly right. You put in oxaloacetate, and again, it increases the oxygen consumption and CO2 production. Okay, so somehow then this is related to oxygen use, but notice there's no place here where oxygen is used, and there's no place here where CO2 is produced, right? So there's all of these reactions are not completely explaining what's going on with the carbon dioxide and oxygen. All right, so it was after this discovered that succinate, we go back up here, succinate is related to a bunch of other organic compounds, and not dicarboxylic acids, but tricarboxylic acid like citrate. Citrate, you notice, is very similar. It's got a bunch of carbons in here with hydrogens on them, but notice it has a carboxyl group here, here, and here. There's three of them. So this is what we call a tricarboxylic acid. 1937, Martius and Knoop published papers that announced this discovery, that you could take succinic acid and produce, I should say you could take citric acid and produce succinic acid with it. And the series of reactions that we now know today, now they didn't show all these details, but the series of reactions we now know is this. Take citrate, you can make it into isocitrate. And all you've done here, notice, the only difference is this OH moves up here. That's it. The OH moves up here and it switches with the H. Everything else is the same. That oxygen there got cut off a little bit. But then we can take this isocitrate and make it into this alpha-ketoglutarate. Now notice, here, 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 this is the same down here, but there's two changes. I've taken two hydrogens off and reflected, like we saw before, a double bond on the oxygen, and notice these two hydrogens end up on NADH. So again, we have an exergonic reaction driving an endergonic one. But notice, how many carboxyl groups are there on alpha-ketoglutarate? There's only two. And the reason is that this carboxyl group is gone. Where did it go? Well, notice carboxyl groups are already almost carbon dioxide, which means the carbon is almost completely oxidized already, which means it's about out of energy. So this carboxyl group gets released as carbon dioxide. Now we finally see a connection between these organic acids and the production of carbon dioxide. Notice there's no oxygen use yet. It's just the carbon dioxide that can be produced. But then the alpha-ketoglutarate can also be made into this thing called succinate, which we saw before. Now there's an intermediate step that Martius and Knoop did not figure out. That happened later. But what happens in cells is this. The alpha-ketoglutarate is converted first to this thing called succinyl coenzyme A. Now it's succinyl because this group right here is essentially succinate, succinic acid. And succinic acid is a dicarboxylic acid, and it's very, very similar to this. But notice there are some slight differences. For example, this other carboxyl group is not really a carboxyl group yet. It has a sulfur bridge to this thing called a coenzyme A. Now, even at the A level here, I'm not going to show you the details of coenzyme A. Coenzyme A is just an organic group that attaches to molecules that ushers them into other sorts of reactions. But once we put this out, notice that one of these carboxyl groups is gone. The carboxyl group comes off again as a carbon dioxide. And we now then have this molecule with still the carboxyl group, which we can then convert into succinate by removing the coenzyme A and replacing it with an oxygen, which is exactly what it does, which again is oxidation, so this gets oxidized. The coenzyme A is released, and it doesn't really do anything except usher these things around. So now this reaction, going from here, succinyl coenzyme A to succinate, is oxidation. You can see that. I gain an oxygen here, so that's oxidation. And that therefore releases energy, which we then use to do this. Now there's a slight mistake in this picture that came from the internet. But here I have a GDP plus a free phosphate to make a GTP. Now remember, we've already seen this before. Adenosine triphosphate is not the only triphosphate nucleotide. There's GTP, TTP, UTP, CTP, all of them. So this is first made in most cells, and that GTP then acts like ATP, except you can't generally use it in your metabolism directly. So what often happens, or at least when we see these kinds of reactions, the GTP then takes its gamma phosphate and donates it to an ATP to make a ATP here. And here's the mistake. You could cross that phosphate out. I don't know why they wrote that, because this GDP plus free phosphate, that free phosphate is not free. It's now an ATP. But here's the point. That the end product is simply this. Succinyl coenzyme A becomes succinate. We release the coenzyme A and we make an ATP. Okay, so here's what we've got now. We've got two carbon dioxides coming off of here, which is great because we produce carbon dioxide when we do this. And we're making NADH here. We're making ATP here, and we can connect it from succinate to these others and make FADH and another NADH here. So all of this is pointing in the direction that this is somehow central to our use of oxygen and production of carbon dioxide in our metabolism. Okay, so this again is the modern understanding of how all these reactions occur. Marcius and Knoop did not do all of that. But this is our starting point for this reaction scheme here. Here's what we know. And again, this is expressed in the modern way. Starting with citrate, we can make isocitrate, we can make alpha-ketoglutarate from that, release the CO2, make NADH. Take an alpha-ketoglutarate, make that into succinyl coenzyme A, again make an NADH into CO2. Take succinyl coenzyme A, make it into succinate, make an ATP out of that, release the coenzyme A, take the succinate, make it into fumarate, that produces FADH2, fumarate to malate, then finally malate to oxaloacetate, and it makes an NADH. Okay, so that's where we were in 1937. So we've got some CO2, great. We don't have the oxygen. We still don't know where the oxygen is coming from. But this is basically our understanding of that scheme when this guy comes along. This gentleman here is Hans Adolf Krebs, and he was a student of a guy named Otto Warburg. If you've ever studied cancer, you'll learn about this thing called the Warburg effect, which is the way that cancer cells use metabolism. And here he is sitting in front of his machine, his pride and joy. Remember I said you could take a bunch of cells and put them into a chamber and measure the amount of CO2 that's produced, the amount of O2 that they're using. That's exactly what this machine does. And nowadays ours look a lot more stunning. They're much more slick and modern looking, but this is what he used to make one of the key discoveries in all of biology. He connected all of that stuff together, and he connected it to the CO2 production. He couldn't connect it to the oxygen. That came much later. But he was able to understand how it is that these organic acids were able to produce all the carbon dioxide.